Mass casualties. Overburdened emergency response systems. Spreading contamination. As the threat of nuclear terror attacks becomes more credible, a serious gap exists in the United States' ability to respond effectively. A radiological event could expose civilians to dangerous levels of ionizing radiation. However, cytogenetic biodosimetry can be used as a proven technique for calculating human radiation exposure during such an event. The Oak Ridge Institute for Science and Education in Oak Ridge, Tennessee, operates a federally funded facility, the Cytogenetic Biodosimetry Laboratory, that can perform this clinical analysis. Cytogenetic biodosimetry identifies damage to chromosomes in individuals who have been exposed to ionizing radiation. It is the most accurate way to determine the absorbed dose, which is critical for physicians managing patients' radiological treatment.
One of the cardinal principles of radiation biology is the fact that the sensitive site within the cell is actually the DNA molecules within the nucleus of the cell. So if we have an efficient method to measure DNA damage within the cell, we can get an estimate of the dose which has been absorbed by the person. Since the chromosomes within the nucleus of the cell are the repository of the DNA molecules, we can study the DNA damage by looking directly at the chromosomes themselves.
The cytogenetic biodosimetry process begins with acquiring a blood sample for analysis. ORAUs can receive the sample at room temperature from any physician's office, hospital, or laboratory around the world as long as it is received within 24 hours. Once the blood sample is received at the laboratory, it is unpacked, inspected, and quickly set up in culture. White blood cells are cultured under sterile conditions in an incubator using a standard growth medium for 48 hours.
One of the disadvantages of the test is that it does take a fair amount of labor, and it takes a three-day period of time in order to come up with the answer. So it's not a quick and easy test, but it is considered the gold standard for biological dosimetry, and it has been proven through the years to be a successful way of estimating the dose with defined levels of probability.
Next, culture tubes are centrifuged and cells are resuspended in a weak salt solution. This allows the chromosomes to separate and spread evenly on slides. The chromosomes are then cleaned and allowed to dry in a controlled environment. Human chromosomes are very small, typically 2 to 10 microns in length, so it is necessary to stain them to enable magnification of up to 1,000 times to examine their structure under a microscope. Once stained, the slides are each mounted with a thin glass cover slip and taken to the cytogenetics workstation. Here, microscopic analysis of large numbers of cells are screened for those specific types of chromosomes that reflect radiation damage.
When we talk about the field of cytogenetic biodosimetry, what we are doing is analyzing the chromosomes, which are the biomarkers of the damage that is sustained. When ionizing radiation passes through the nucleus of the cell, it induces what are called double-strand DNA breaks. If those breaks are not repaired properly, it can lead to an exchange-type chromosome aberration and form what is called a dicentric chromosome, or a chromosome with 2 or more centromeres on it. As it turns out, the dicentric chromosome frequency is a sensitive index of the amount of radiation that's passed through the nucleus of the cell. So in cytogenetic biodosimetry, we're primarily concerned with the measurement of those dicentric chromosomes.
Dicentric chromosomes are located using an automated workstation consisting of a highly sophisticated microscope coupled with a slide changer. The microscope can automatically process as many as 80 slides unattended overnight. Each slide, which can hold up to 1,500 dividing blood cells, takes only 6 minutes to process. The microscope software can create a karyotype table, which allows researchers to quickly identify chromosome abnormalities. The frequency of dicentric chromosomes is then compared to a dose-response calibration curve to identify the type of radiation a patient has been exposed to, as well as his or her estimated dose. Only the Armed Forces Radiobiology Research Institute in Bethesda, Maryland can provide a similar service in the United States. However, its resources are dedicated to military applications of the technology. The ORAU lab also participates in an international biodosimetry network along with 4 other laboratories. Researchers continue to develop methods to improve the accuracy and speed of cytogenetic analytical methods.